welcome back to Davin Biology. So as you can see in the topic title of this video, of course I'll be going through Kerja Amali lah to prepare you for your tomorrow exam. Yeah, so by the time this video come out, it will be less than 24 hours before you sit for your Kerja Amali. Now, I might not sound very well or look very well in this video. That's because I just got COVID. Yeah, but don't worry, I'm fine. Still can do video for you all. Okay, so come. Now, I'm sure you all receive a lot of a lot of tips, 有时候风对不对 whereby the most highest probability punya experiment that's going to come out for bio is your Form 4 Chapter 7, Fermentation in Yeast, Fermentasi dalam yeast. Okay, which is actually alcohol fermentation, fermentasi alcohol. Now, in this experiment, now every time remember when we do Kerja Amali, right? The first thing I want you to find out is the variables. 先去找那个 pembolic ubah. Okay, so I already give you here. So by the way, yeah, this set of notes I will put a soft copy in the description below. Okay, you can go and find it. So here I already prepared like example questions that they could ask, and also I already prepared example answer already. Got BM and both English. So for MV, manipulated variable, first is the presence of yeast. Okay, so you can see in the diagram there are two set up. One have yeast, one don't have yeast. Now for the bergerak balas responding variable finding simpian, so there'll be a few possible different responding variable. Now I cannot confirm with you or pinpoint which one will we be using in your actual exam. Now it depends on how your exam asks. Okay, I will explain more later lah. Okay, so let me just briefly go through like how do you set up this whole experiment first while you're doing it. Okay, so you can see we have two setup over here, two conical flasks A and B, kelalan cone A and B. Now obviously the main difference here is one you have yeast, one is just distilled water. So one is glucose and yeast, one is glucose and distilled water. Of course, the one with yeast is going to carry out fermentation, alcohol fermentation. Okay, so yep, remember the equation for fermentation, alcohol fermentation is glucose, arrow, CO2, ethanol, and energy. So the way we detect whether there is alcohol fermentation, right, is your responding variable. So for example, you can see in this setup, they connect a tube here to a lime water. So lime water, you all should know by now, is to test presence of carbon dioxide. So if there is fermentation, alcohol fermentation, there will be carbon dioxide produced, and obviously your lime water will become cloudy. Air kapur menjadi keruh. However, this one, there will be no reaction, no fermentation. So therefore, this lime water will remain the same. So therefore, the first type of responding variable will be this one, condition of your lime water, keadaan air kapur. Okay, now however, I got received some of the tips from my students telling me that instead of using air kapur, they might use something called bicarbonate indicator, penunjuk bicarbonate. Now, this thing is also used to detect the presence of carbon dioxide. Okay, actually, it's more commonly to detect the changes of pH. But since carbon dioxide is acidic, they can pick up the presence of carbon dioxide. So instead of lime water, they will put the bicarbonate indicator over here. So if there is fermentation, right, this thing should turn yellow. So basically, you can see your bergerak balas, finding simpian soup, is final color of bicarbonate indicator. And another possible responding variable is also your final temperature. Now, that's because fermentation releases energy that's heat. So this one will be slightly more easier. All you have to do is just put a thermometer inside here, record the initial and the final temperature. So at the end of the experiment, your temperature should increase 1 or 2 degrees. Okay, now, so I'm probably sure you all got hear like experiment, you have guided and unguided experiment. So last year SPM came out guided. There was procedure, there was everything, all the steps listed down nicely. However, this year there might be a chance that maybe, I don't know, they might ask unguided. So let's say if they ask you unguided experiment, right, definitely you will need to write the procedure. So procedure will be 4 marks. Now a lot of you will like, how to get these 4 marks? How do I write so much to get 4 marks? Okay, actually just let me tell you, These 4 marks, right, in order to get it, right, your procedure, they always will say your procedure should include method to handle variables and also precautionary step. Cara mengenali pemboleh ubah and the langkah untuk berjaga-jaga. So you have 3 variables, so that will be your 3 marks. You must mention how you handle the 3 variables plus 1 precautionary step. So that's how you get the 4 marks. So actually in this procedure over here, now let me tell you where the marks come from. Okay, so you can see the first one is actually the constant variable. Boleh ubah dimalarkan. So the volume of glucose, or you can say the concentration of glucose. So both, I will put the same amount of glucose. Okay, I mix with 100 ml of distilled water. Now you can also say this, distilled water volume. Okay, now the next one, okay, boil the mixture and cool it at room temperature. Now this step actually we don't have to carry out one. I think most likely this step they already boil for you already one. So we are not required to boil the glucose. Yep, so this is your langkah berjaga-jaga precautionary step. Now, why do you need to boil the glucose? Because boiling the glucose will remove oxygen. Because remember, this is alcohol fermentation. So we need to boil the glucose to remove the oxygen. So that there's no oxygen, then there will be fermentation instead of respiration. Alright, so the next 2 points, this is actually your manipulated variable. So right here first, this is your constant variable. Dimalarkan. Okay, and this 2 is your MV. Okay, right here PUD lah, dimanipulasi. So remember your glucose, the presence of yeast is our MV, correct? So yeah, you can see 1 conical flask, I put yeast, 1 I put distilled water. So long you mention your MV, what is the MV that we use, which is yeast and water, you mention these 2, then you got marks for MV already. Okay, then I add paraffin oil in both conical flasks. So why I need to put paraffin oil? This is to prevent oxygen to dissolve in the solution, because remember we are carrying out fermentation, we don't want oxygen. Okay, then I leave the apparatus for around 15 minutes. So I will let the fermentation carry for 15 minutes and then measure and record the initial and final temperature of mixture using thermometer. Now it depends on what is your responding variable. So look closely on what the apparatus they give you. If you could give you instruction, read carefully. So let's say if my responding variable is temperature, then I will say measure and record using thermometer. So measure and record your responding variable. Okay, if let's say if lime water you cannot measure lah. So we will say observe and record. So just now was mengukur dan merekod. Now it's memerhati dan merekod keadaan air kapur, because you're going to use your eyes to see the lime water. Okay, they use eyes to see, then you record. So this is observe, memerhati, 观察. And same with bicarbonate indicator. This one also use eyes to see the color changes. So therefore, it's also observe and record final color of bicarbonate indicator. So your responding variable, PUBB, bergerak balas, is always at the last point one. Okay, let me just highlight all the points here. This is dimanipulasi, MV. And last one, this is your responding variable. Okay, so this 4 sentences will give you your 4 marks. So actually, you can just write this 4 sentences, then you get 4 marks already. You don't have to write all the other things also can. Okay, now, then here is a table where they ask you to record your observation. So once again, depending on what is your responding variable, you record different kind of observation. So yeah, if it's changes in lime water, of course, this one got fermentation, will become cloudy, this one will remain clear. Okay, smell, I don't think we're going to use this one. Okay, so temperature, you can see 1 increase, 1 remain the same. So if it's the bicarbonate indicator, 1 you can say turns yellow, 1 remain unchanged. Okay, so it's bicarbonate indicator, 1 will turn yellow, 1 unchanged. Okay, right. Then let me just go through a few example questions they might ask. So sometimes they will like to ask variables. Very easy, right? Variables boleh ubah. 我们刚才已经讲了. Okay, next. Now the questions here you can see is related to some of the questions related to the knowledge of your knowledge regarding the experiment. Like just now we mentioned already, why glucose solution need to be boiled? Mengapa larutan glucose perlu dididihkan? 为什么你要滚那个 glucose? I already told you just now, that is to remove dissolve oxygen untuk meningkatkan oxygen terlarut. Okay, 1 mark, easy. Now state the function of this substance. So lime water. So lime water is to test the presence of CO2 lor. Okay, if let's say they say bicarbonate indicator, then also the same is to test presence of carbon dioxide. Penunjuk bicarbonate also the same. Okay, then paraffin oil just now I also got explain to you the law. It prevents oxygen from dissolving, menghalang udara daripada melarut, or you can still just say oxygen. Alright. Okay, D, inference. Now, if you don't know what's inference, inference is basically explain your observation. Okay, so for example, if your observation is this one, lime water, why this one the lime water turn cloudy, why this one remain clear? Or let's say your exam is this one, bicarbonate indicator, why it turn yellow and this one remain unchanged? So now very easy. So all you have to explain is because this one turn cloudy, this one turn yellow because got fermentation. No? If got fermentation, of course you release carbon dioxide. So it depends on what's your responding variable. So if let's say it's these two, we can just say because in A got alcohol fermentation, release CO2. That's the main one. If it's temperature, then you must say energy. Okay, so out of these 3 products from the fermentation, depending on what the responding variable is, you just have to state either one good enough already. Or you can just write all the 3 products of the fermentation. Also can. Okay, right. Then, so BM1 also the same lah. Fermentasi alkohol berlaku menghasilkan CO2. Now for B, no alcohol fermentation. Fermentasi alkohol tidak berlaku. That's why no CO2, no energy. That's why no CO2, your lime water remain the same. Bicarbonate indicator does not change. Temperature also does not change. Okay. Now the last question here, hypothesis. Okay, hypothesis here, nothing much. Now this hypothesis, right, we don't use the yue, means the higher the MV, the higher the RV. Now this question, the hypothesis is a bit different. So in this experiment, you can see both conical flask A and B, right? Inside got no oxygen. Okay, so what I want to test here is when there's no oxygen, what will happen to my yeast? So before I carry out the experiment, I come up with a hypothesis. We'll make a smart guess, 做一个假设. So I will say in the absence of oxygen, yeast undergo fermentation. Actually must say alcohol fermentation. So the B1 also same, fermentasi alcohol. Okay, which produce the 3 things of ethanol, energy, carbon dioxide. So that is how we write the hypothesis for this question. Okay, so once again, remember, observation usually will be just record your result in the table, which is easy marks. Then variables, this one you know lah. Then remember lah, inference is explain your observation. And for fermentation, these are the type of questions that they might ask. It's very simple. Okay, I also got prepared another experiment. Okay, over here, in case this one can come up, because I also see some of the tips got this one. Okay, so another wind also got blow this one to me and tell me this one maybe got chance. So this is your Form 4 Chapter 3. Movement of substance across semi-permeable membrane. Pergerakan bahan melintasi membrane telak memilik. So this is the whisking tube experiment. Okay, let me just go through the whole thing. Now, all you're going to have here is you're going to have 2 whisking tube. Okay, and I'm going to soak it in 2 different beaker with distilled water inside here. Okay, so inside here is distilled water. Okay, water. Now, in these 2 whisking tube, one of them, definitely I put starch, kanji in BM, and the other one is glucosa. So between these 2 molecules, of course, glucose is the small molecule that can pass through the whisking tube. Starch definitely cannot, because starch is polysaccharide. You should know this by now. Glucose is a mono. So for this experiment, we're going to test what kind of molecules can pass through semi-permeable membrane. So therefore, this experiment, your MV, pemboleh ubah dimanipulasi, is size molecule. Okay, I didn't have answer here. Okay, I just write here. Size molecule. So your glucosa and kanji. Molecule size. Size of molecule. Okay, now your responding variable, is like this. So your responding variable, you need to know how to carry the experiment. Then only you know your responding variable. So for kanji here, I will put the water here, right? Okay, after let's say, okay, I will soak this like, let's say around 15 minutes like that. 15 minutes later, right, I will take out the water from here. Now for kanji, I will mix with iodine. Because iodine is to test presence of starch. For this one, I will put it in Larutan Benedict solution. Because Benedict is to test presence of glucose. So in the end, your iodine will remain yellow. Here, remain yellow. Because there's no starch that will come out. So if got starch, yellow will become blue-black. But of course, this one will remain yellow because starch cannot come out. Benedict will turn from blue to brick red precipitate. Biru kepada mendakan merah bata. Okay, but however, right, for the Benedict one, once you test with the Benedict already, right, you need to boil it. You need to didihkan the larutan. Then only you will have these changes. So if they come out this experiment, this is what you need to boil the solution. That's why I think this one, the chances slightly lower because the step actually very long. Okay, so the first question, okay, this is unguided. I give you the procedure here. Your procedure, same thing, is to include method to handle variables and precautionary step. You can see the PM1 and next page also the same. Cara mengendalikan pembalik ubah dan langkah berjaga-jaga. Okay, so where are all the three variables first? Okay, so the first point is your constant variable, 10 cm. So your whisking tube, length of whisking tube, panjang tube whisking. So you cannot be more than 10 cm. I fixed it at 10 cm. So the volume inside is the same. Okay. Then I pour 10 ml of glucose and starch into two different whisking tubes. So this is basically your demanipulasi. Because size of molecule, what are the size of molecule I want to test? I'm testing glucose and starch. Glucosa dengan kanji. Now this is your langkah berjaga-jaga precautionary step. So once I fill the whisking tube with glucose and starch already, I will need to rinse it with running water so that I wash the excess, the glucose or starch that spilled outside on the whisking tube. Because if not, then the starch might enter here, the glucose might enter here and change your result of the experiment. Okay, then place both whisking tube into a beaker filled with distilled water, right, over here, beaker filled with distilled water. And I leave it for around 15 minutes. Okay, then, like I said just now, both I take the water sample, starch one mixed with iodine, glucose one mixed with Benedict. Here you got say. The glucose one, I mix with Benedict. The distilled water from the beaker containing starch, I mix with iodine solution. Okay, but of course this one you have to boil it lah. But here I didn't mention because you mentioned boil the glucose, boil the Benedict got no marks one because that is not the variables. So actually you don't write boil, actually also no marks one. That's why here I leave it out. Okay, and then the last point is your responding variable. Bergerak bala. So bergerak bala is always behind because responding variable is always what you get at the end of the experiment. So at the end, this one also don't need measure and record, just observe and record the final color. So use your eyes to observe the final color. Okay, yeah, so that is procedure lah. Now, then let's move on to example questions here. Okay, so here, state your observation when distilled water outside the whisking tube is tested with iodine solution at the end of experiment. So the one that I test with iodine, the starch one. So we already know starch, I say, was large molecule. It cannot diffuse through the whisking tube. So for iodine one, it should remain yellow. Okay? Or you can say brownish yellow. Kekal kuning keperangan. Okay? Right. Then, next, explain your observation. So explain your observation is same as inference. Okay? So don't get confused. So just like just now, we need to give a reasoning behind your observation. So why your iodine does not change? Because I already told you, starch is not found in the water, right? Because starch cannot diffuse through whisking tube due to its molecule too large. Tidak dapat meresap melalui tube whisking kerana size molekulnya besar. That's the 2 marks. Cannot diffuse because large molecule. Okay, then here is the result of experiment. Write your observation in the space provided to show the ability of molecules diffuse through the Visking tube. Now, I don't think in the exam they will give you so many things to write. I think at most, right, it's just bahan, size, and the color change. Maybe these 3 only, I think the most. Let me see. Ability of molecules to diffuse through. Yeah, should be around this few only lah. Maybe you have to write the availability lah. Yes or no, can or cannot. So glucose can, starch cannot loh. Glucose boleh, kanji tidak boleh. Okay, so the one that boleh of course is kecil. The one that tidak boleh of course is besar. So the color change you already know loh. For glucose one, the Benedict, biru to mendakan merah beta. And kuning will remain kuning. Brownish yellow will remain brownish yellow. Okay, then next, explain your observation. So what we observe here is the glucose can change the Benedict color, but the starch does not change the iodine. So it's the same as what we wrote over here. Okay, that's because okay, Benedict, this one they want Benedict. So it's the opposite of just now. This one's iodine, starch cannot, right? So this one glucose can. That's because can diffuse through Visking tube. Molecules are small. So these two is the main point. This sentence actually is the main one. You can pass through Visking tube because molecule is small, big. Understand? Now, last question. So Ramalan, prediction question. So they say, now I replace the glucose with maltose. Okay, so maltose is a disaccharide. So disaccharide actually cannot pass through the Visking tube one. Now, so for this one, my prediction is my Benedict will remain blue. Yeah, so by the way, Benedict can test presence of all your monosaccharide, girlfriend, gorgeous, glucose, fructose, galactose, and it also can test presence of 2 of your disaccharide, maltose and lactose. Sucrose cannot lah. Okay, so maltose, if it comes out, your solution will become red precipitate. However, maltose is disaccharide, it cannot come out. So therefore, the distilled water that you test with your Benedict won't have any changes. So it's the same like what we wrote for starch just now. They cannot diffuse through Visking tube because molecules are large. Tidak dapat meresap melalui tube Visking and size moleculenya kerana size moleculenya terlalu besar. Okay, so that's it lah for this video. So by the way, these are the 2 experiments that what I received lah. The highest probability that these 2 experiments will come out. Okay, so once again, yeah, you can download the file. The link is in the description below lah. So good luck for your exam, for your Kerja Amali, and the SBP paper is coming out very soon. Okay, so I will see you guys in that video lah. Bye bye. 新年快乐。